of nursing. So I'm in the happy mom spirit. The next, the next to that is I learned with IDG, which I think a few people know about in here. And I learned with IDG resonates with the story of Dr. Fala from yesterday. When I started my PhD, my molecular biology was from a distance. Okay, I was privileged to attend maybe 2 practical sessions where, with my height, it doesn't help. So there is a group of 10 people, and I'm there trying to see who is holding the pipette, who is behind. That was as close as I went to to about molecular biology. I started the first PhD in Nigeria here. I was way into my 2nd year, starting my 3rd year. I am somebody who is always hungry for the new thing, something stimulating. So I decided to abandon that PhD and restart. And I restarted in PhD molecular biology and biotechnology, big name. I still remember my very first class, and it was at the niche of the lockdown. So I resumed in February, the lockdown commenced in March. So a new environment, new people, new experience, everything happening in my cubicle. It wasn't even the standard size of a room. So the first class, there was introduction. What do you do? What have you done with with molecular biology? Then my colleagues went, oh, I was in Germany. I did this, synthesis, CRISPR. I'm like, whoosh. I'm in the wrong room. Yeah, I was definitely in the wrong room because all I said was my name and I have a background in microbiology. Then they're talking about SNPs. They were talking about CRISPR. And we were just 11 in class, so there was no hiding place. Then I remember calling my mentor after that class, sir, I'm coming back to Nigeria because I was my local champion. So let's maintain the energy. But he said one thing, I do not know you to be a quitter. So you have 2 options, take the easiest slot to fly back home or prove the reason why you are who you are. So I took the rest of the day to cry because I do that a lot. And the next class, I decided to change something. And then I answered a stupid question. And that even made the embarrassment, to the roof. But every time I had the moment, I was so grateful for the lockdown then because I had nothing else to do. I wake up, I'm reading. I get a minimum of maybe a couple of sleep, then I'm reading again. And that process helped me to come up to demystify the ambiguity of molecular biology. Sometimes the technical jargons are so scary. You hear the big names and it's like, that is meant for magicians. It can never be me. You don't have to be Merlin, okay, before you understand these things. And that is what I learned with IDG is doing, to simplify molecular biology techniques to as simple as possible for people across Africa. And we've been doing that for over 6 years now. And it's free. And then the third one there is the Women in STEM Support Hub, WISH. WISH is specifically for women. Sorry, majority of what I'm saying says girls, women, girls, women. I learned with IDG is open to all. So WISH is for women because I noticed that majority of women start the postgraduate program, but they don't finish it. Or they finish it much later than their male counterparts because there is always the commitment of womanhood, but we also need them to become excellent scientists. And for the past 3 to going to 4 years now, I started this with one of my PhD advisors actually, and I'm so proud to be able to do that with her. And lastly, which is the baby of them, is the AfriHealth Public Awareness Consortia, which is something that also stems from what I found to be a problem in the society, which is there are a whole lot of public health concerns. But then when they are delivered as a campaign, it is in the quaintest of English. Then how do you want women with 0 literacy who sells something by the roadside to understand the information you're trying to pass about basic things like antimicrobial resistance, like HPV screening? So APAC is working with amazing people across Africa to develop public health awareness campaigns in indigenous languages across Africa. So we have Kiswahili, we have Luyanda, we have so many languages that we are working on to deliver this. Why is this slide important, you might ask? Everything on this slide is the reason why I work in the lab. My beyond the lab is the stimulation motivation, because if you do full-time research like the established Ross and Yumi can say, you get exhausted. There are days where I question this research. I mean, I can just be a baby girl and do something else. But there must be something that keeps you on your toes, something that takes you back to the lab the next day. And faces like that smile and send me videos at 10 p.m., that's the reason why I stay in the lab. Now to ICGB, the International Center for Genetic Engineering and Biotechnology, who are one of the sponsors for this amazing workshop. Last week for the neuro track, we had the scientific coordinator with us, Emanuele Burati, and he was able to share more about ICGB, so I won't be doing his job. But I'll just tell you briefly, ICGB is an international organization for research, training, and technology transfer in life sciences. And the sole goal is to be able to promote sustainable development. We have three main components across the globe. The headquarters, which is in Trieste, Italy. We have the component in New Delhi, India, and in Cape Town, South Africa. I have been working at ICGB for about 4 years now as a postdoctoral researcher in the tumor virology lab under the leadership of Dr. Lawrence Banks, and he also doubles as the Director General. So we send you greetings. And there are different research areas in ICGB across the components. So please feel free to visit our websites to explore opportunities on fellowships, grants, collaborations that might be useful for your work. And we are also very active on social media pages. Now, disclaimer, I don't work with Drosophila every day, but I have worked with Drosophila and I still work with Drosophila. I'm privileged to be part of a charity organization that is fostering the use of Drosophila as an ethically viable and affordable model across Africa, the Dros Africa. We had Dr. Isabel and Professor Lola, which are part of the founding members last week, and they shared more about this. So please, again, go to the website and read more about how you can contribute and actually benefit from Dros Africa. And then to what I have done with Drosophila. Here is my first baby project on the use of Drosophila, and just a short story. Until 2023, when I met Professor Abolaji, I have heard about Drosophila, but I so much about Drosophila, right? But then his talk was on Risabatol. And then what caught my attention was actually in the anatomy of the fly. My mind is always in recombinant expression of protein because I love therapeutic peptides. A lot of people do plant work. I understand where you're coming from. Antibacterial. From 2012 until 2019, I did antibacterial activity of something against something. I've moved from mango to moringa to several things. I mean, I've been there, a lot of plants. But one thing that hurts me is, so what? And the so what has always been missing. So we chunk out a lot of papers, antibacterial activity of mango. Okay, so what? Eat more mangoes? Super. But then something was missing. And my PhD formed the link for me. Forgot to mention that moving from zero in molecular biology, within 2 years into my PhD, I developed 2 patentable products. And that is to tell you, you can always go from negative, because I wasn't on zero. I was on negative 100. And I moved to 1. I wouldn't say I moved to 100. I moved to 1 because I'm just starting out. So from his talk, I'm like, these things are so fascinating. I'm looking at, so if I'm able to use the fat body, which I read more about, which is a natural site for antimicrobial peptide synthesis in the fly. What if I put my difficult to express peptides in the fat body? What will the fly say? And that was this project, which was generously funded by the Cambridge Africa Alborada Grant, and in collaboration with Professor Tahir Okin from the Department of Genetics, Cambridge, an amazing, amazing man to learn from. So we did that, and I was able to successfully express 2 therapeutic peptides, which I've confirmed before in E. coli, in Drosophila. And I remember sounding crazy to a lot of people when I got to the Department of Genetics. Like, oh, okay, do you think it will work? I don't know. I mean, if I'm crazy to come here, we might as well find out. And everybody kept following up. So what do you see today, Ife? I don't know, I'll let you know. And eventually we got those peptides running, 2 of them nicely expressed. And we've been in communication with the Cambridge Enterprise, and we are following the consultancy pathway for technology transfer. So not only were they were expressed in good chunk, even at larva stage. And they retained activity even through all the process, the biological activity of these peptides are retained. And right now we are planning on how to make the SoFly BioWorks, which will be incorporated into the DRTC technology transfer platform. So it's about big proteins, tiny flies. And that is what I am doing currently with Drosophila. It's not cancer, but it's still providing something that can cure cancer. Mindset shift. Now, to talk about why do infections matter in cancer, perhaps we need to focus on the fact that 15 to 20% of cancers are attributable to infections, okay? And this prevalence, this number is spread across the globe. It's not restricted to one geographical area. You have more prevalence in Africa, in Asia, and some parts of the Americas, but it is everywhere. Now, if you have a closer look at my favorite part of the continent, if you look at Africa as a continent, then you see these numbers even going higher. In some parts of South Africa, in some parts of East Africa, in some parts of North Africa, you have more than 37.6 events per 100,000 population in 1 year. So it's closer to home than we think. Now, who are the major players? How do infections cause cancer? The four major guys that are involved in cancers attributed to infections are Helicobacter pylori, H. pylori, that came up a lot last week. HPV, human papillomavirus, which is what my research is on, hepatitis B virus, hepatitis C virus. And majority of this has high prevalence in the African setting. A lot of people study gastric ulcer, yeah. Ulcer. But what is the biology of the ulcer you're studying and that you're giving all these amazing plant extracts to? In Africa, in Oceania, the Americas, HPV actually is attributable to majority of the cancer cases that stimulate or that is associated with infections. We see the huge number there, about 850,000 cases globally just for H. pylori. And for HPV, about 730,000. So it is something that needs a lot of attention. And how can we study this? The data I'll be presenting here, we're not done in Drosophila. Like I mentioned, I don't do Drosophila every day. But something that got reiterated over and over last week is that there is no optimal model. You can't answer every question in one model. I can pick my cell line and I have limitations. Depending on the context of the cancer I'm working with, like HPV, for instance, you cannot recapitulate the natural infection in flies. Why? They have a high host specificity. So you can do HPV. We do have a lab that has started working on trying to mimic HPV oncogenesis in fly, but it's still one lab. And we hope to hopefully see more. So what are the basic hallmarks of pathogen oncogenesis? If you look at HPV, H. pylori, or cancers that are even not from communicable or infectious agents, certain things are common to them. You have cases of inflammation. So chronic inflammation, oxidative phosphorylation, mutations. Now, which of these partners, if you think about H. pylori, please read more about it. I'm not an expert in it. But the CAG-A protein is a major virulence factor that induces chronic inflammation. So what happens with inflammation? Back to our basic biology. You have infection in the site, you have chronic inflammation, you have recruitment of immune cells to that site, right? And that triggers the accumulation of reactive oxygen species and a lot of cascade of effects that eventually leads to genomic instability, and then you begin to accumulate or accrue a lot of mutations. So you have one thing starting it, infection. But not just infection, because the immune system tends to clear majority of this infection else. I guess majority of people in Africa would probably be down with gastric cancer by now. But persistent infection is something peculiar to oncogenic pathogens. They're able to stay within the body, evade immune system. And my favorite model is the guy I work with, HPV. That's one very chilled guy that knows how to evade the immune system. So that's him there, or she. And okay, fair. And this guy knows how to avoid immune system. First thing is the silent replication. HPV goes to the basal layer of the epithelium and keeps his own gene expression very quiet. I don't want the body to know I'm here. Cervical cancer or any cancer associated with HPV does not happen in 2 years. It does not happen in 3, not even in 10. It can stay silent in the body for decades, silently changing the integrity of the epithelium until the damage is caused. So it is asymptomatic. And HPV is the most common intimately transmitted infection in the world. Little wonder, because it can stay quiet within the body. And aside just staying there, replicating just minimally, it is also able to manipulate the antigen presentation mechanism. So it goes to MHC1, say, hi, I want to stay here, let's work together. I reduce your expression, you don't expose me. It does that a lot. So MHC1 is down, antigen presentation is down at the same time. So you lose bringing cytotoxic T cells to that site. And it's not doing this one after the other, it's concurrent. Another mechanism that it uses is to be able to release interleukin 23. And when you have a burst of interleukin 23, what it does is it marks the proliferation of T cells. So this guy is able to use multiple mechanisms, which is something very peculiar with pathogens. Multiple mechanisms to achieve one goal. Do not kill me before I kill you. That's it, pretty simple. And why am I going into the details of this? A lot of people yesterday during the introduction mentioned, I'm interested in immunology. I'm interested in this, I'm interested in that. You can always pick one baseline and study multiple things under that umbrella. We can all sit down with H. pylori, HPV. I am working on the epigenetic part of things. You choose to work on the immunology part of things. Then we have collaborative research that helps us develop better research, not just individual chunking out. You know, Africa is so interesting that this is how we do science. Do you get what I mean by that? In isolation. When you hear a knock on your door, close my laptop. Science is so, it should be open. It's easier when you collaborate. It reduces, I mean, you check the immunology, or you are an expert in checking maybe the physiology of things. Give me the data from that. We come together and we build something strong. It's more believable because you have a cohesive, complete story. Just driving a shift of mindset. Another thing that pathogens do is to disrupt the apoptotic pathway. This is very common. Apoptosis, right? I would also use HPV again because we've gotten to be close friends now. And this guy has two major oncoproteins, E6 and E7, and they work side by side. So E6 says, okay, now we found the cell. I'm going to go mate the ubiquitin ligase E3, and I take it to P53 to continue to polyubiquitinate it and degrade it so that we override the P53 mediated apoptosis. Then E7 says, okay, bro, I'm just going to go down to PRB, inactivate PRB, and override the controlled cell cycle arrest there. So doing two things concurrently, you shut down apoptosis, you shut down controlled cell cycle arrest, and down the cell's guard is down. And in one of my recent papers, I was able to find that E7 and PRB actually have like a chaperone activity. So when you don't have PRB, E7 stability goes down. So not only is it a host substrate, but it needs it to be able to remain within the cell as well. And that was a very interesting study by that, because I did it. And the epigenetic reprogramming is also a hallmark, one major hallmark of pathogen oncogenesis. So sometimes the DNA is not changing. There is no actual mutation in the sequence, but there are chemical modifications on the histone. You have methylation, like our ongoing work on PRMT, which is a methyltransferase. And what you see is that when methylation happens with P53, E6 also uses the methylation process, actually, to ensure that P53 is exposed for more degradation. So the methylation supports stability of P53, but E6 blocks that. Say, PRMT1, come methylate me, actually, and make me stronger instead of doing it for P53. So it's thinking about understanding the biology of your etiology. And with that, you can ask so many research questions and find innovative ways to answer them. And this is what I will be talking about today, which is the core of my current ongoing research, and is the disruption of polarity and tissue mechanisms. So I was speaking with, I think somebody yesterday talked about membrane proteins. I work with a lot of membrane proteins now, and we are looking at how the tissue mechanisms and the polarity is actually hijacked by HPV. And that's what we'll be looking into. We already saw this earlier, that HPV is a big deal. And the prevalence of the high risk, because we have the low risk, which are non-cancer causing, And we have the high risk, which are cancer causing, okay? And then you have the distribution, the prevalence is quite global, but then look at the number here, right? Another interesting thing is that the subtype that is prevalent tends to vary across the globe. But one guy is consistent across this number, and that's HPV-16. Is the most prevalent that you find in cervical carcinomas. And if you look at the incidence here, cervical cancer still ranks as number 8 of 10 most occurring cancer cases worldwide. And the mortality rate is still equally high. So there is still a need to understand HPV biology because currently there is no treatment that stops HPV. What you can do is manage cervical cancer if detected early. So like I said, just a brief of how HPV works. Its favorite natural site is the basal layer of the epithelium. So HPV comes, infects the cells here, but at that time it's just maintaining its own genome. No noise, no immune trigger. Then its life cycle follows the natural differentiation of the epithelium. So as the layers of the cells are maturing, it grows with it as well. So the cell grows it as part of itself. No drama, right? So when it gets to the suprabasal layer where you expect cells to move out of the cell cycle, E6 and E7, remember the P53, PRB, they help it to override the cell cycle checkpoint and keep the cells in active cell cycle dividing. That is the hallmark of HPV oncogenesis. And after that, the virus assembly occurs, and then the shedding is not dramatic. You know, when the skin sheds naturally, the mature virions also go with it. So it knows how to stay silent. And that's it. There, the genome. Now, my favorite oncoprotein to work with is E7. Why? Because it's a very small guy, about 110 amino acid. But if you look at the residues here, almost every residue here has a host substrate that it binds to. And that is why our lab prefer to work from the proteomic side of things. I have a strong bias for proteins. Why? Any guess? Why proteins are not genes? Sorry? Thank you. They are the actual effectors. Is the last thing you see. There are so many things that you can capture, or gene differences, differences in regulation of the genes, but they might not be captured at translational level. And one other thing is that something happens to proteins after translation. What is that? Super. Post-translational modifications. Can you see that at gene level? So that is why we prefer to look at things from the proteomics aspect of things. We want to see what is happening to the active cell state, not what is being predicted or likely to happen. Well, I hope that was a good argument. So looking at E7, there is ongoing work that helps you to identify tons and tons of host substrates that E7 interacts with. And our lab has contributed to a lot there. Today, I will be focusing on the CR2 region. And this is a very interesting region, just less than 30 amino acids, but this is where the damage occurs. This is the region that has the motive for PRB binding, our recently identified adaptor protein molecule complex, which is used for endocytic clathrin-mediated endocytosis. And then we have these two serine patch here, which is known as the casein kinase phosphorylation site. Now, this is where the story changes. What we know is that CK2 phosphorylation enhances the oncogenic potential of E7. So if it's already a bad guy, when it becomes phosphorylated, it becomes even a badder guy. So we are much more interested in the lab because we've seen that when E7 is phosphorylated, it degrades RB even more, faster, and more efficiently. We know that it can only control clathrin-mediated endocytosis, which affects internalization of EGFR, by the way, when it is in its phosphorylated form, and many others. So our constant interest is to identify specific interactors of CK2 phosphorylated E7. So how do we answer this? I hope my talk today will do one thing. Help you to think about approaches to asking and answering research questions. This is all I want to achieve, and I hope I can do that with one person. So to do this, the simple thing we did was to design peptides. We synthesize peptides that correspond to the region I showed earlier, the CR2 region. And we did that with phosphorylating them or not phosphorylating them. So what happens from here was you take lysate from the normal keratinocyte, which is the natural site of infection, and you incubate it with these peptides. You have an immune precipitation whereby, when you have, if there is a protein-protein interaction, it will bind to that peptide. Then you analyze the immune complex by mass spectrometry. Simple. And from there, we found VANGO1 to be specifically bound to phosphorylated E7. And VANGO1 stood out because we know that E6 is the oncoprotein that has affinity to polarity, or to PBM, which is the PDZ domain containing proteins, not E7. So seeing E7 bind us to VANGO1 was quite intriguing. And because we cannot just take this and run with it, we had to validate it by immunoblotting. Majority of what you'll be seeing in my results are Western blots. It's our thing. So we did find that indeed VANGO1 is selectively binding to the phosphorylated form of E7, but PRB can bind to both. But VANGO1 stays just with the phosphorylated one. So we wanted to see what was happening. You asked from the protein. Now, how do you confirm the relevance of this? What we did was to pick several proteins, E7 proteins from different HPV subtypes, and do what you call an in vitro protein-to-protein binding. Very simple assay. Immobilize one protein on beads, a solid substrate, add another protein in solution to it, incubate them together and run your gel, and probe for that protein. If there was a protein-protein interaction, the blot will show you if it's happening in vitro. Simple assays to answer important questions. So we found that, yes, VANGO1 actually prefers to bind to HPV16 compared to other subtypes, high risk, low risk, or the cutaneous one. And sometimes when you do a lot of protein work, you know that some interactions happen in vitro, but they are not recapitulated in vivo. So how do you ensure that? Another simple assay, whole immunoprecipitation. In simple words, I have one guy, I put flag on it. I have another guy, I put HA on it. I mix them together. If I want to confirm whether flag interacts with the HA1, I use flag to pull it in, then I probe to find the HA. Do you get it? It's like you have just one bait to fish, and you want to catch a crab and a fish. You know that sometimes the fish will swallow the crab. So you target the fish with the hope that you find the crab in its gut. So that is how co-immunoprecipitation works. And that's what we did. And we found that when you pull down, E7 flag also comes down with it. Flag VANGO1 also comes down with it. So now we know that this interaction happens in vivo, so it matters. Then the next thing was, what is the importance of phosphorylation in this mix? Simple thing, create a phosphomimic, right? And all you need to do is just switch one amino acid for the other, site-directed mutagenesis, that you can do with simple PCR and designing your own primers. No magic. So we switched serine for aspartic acid and realized that phosphomimicry is not sufficient for that interaction to happen. So what does that tell me? A true phosphorylation event by CK2 is actually needed. Check, phosphorylation is needed. And how important is that? Now, everything we do has an endpoint, clinical importance. So for us to be able to know if VANGO1 is even worth studying and expending all these resources on, we know that currently there is a pharmacological inhibitor, CX4945, that is targeting casein kinase 2 activity, the CK2 activity. So what we did was to treat the cells expressing growth proteins with this inhibitor. And we realized that indeed, E7 binds less VANGO when you inhibit that CK2 pathway. So it might be a pharmacologically relevant pathway to pursue another interest. So we can move forward. Now, what is VANGO1 and why should we study it? VANGO1 is a core component of the Wnt planar cell polarity signaling. there is usually a lot of attention on the apical basal polarity, but what happens in the plane, the middle of it? Yeah, this is the pathway that directs the cell orientation and cell division, yeah, in an oriented manner. Think about it this way. So you're thinking of building a house. You make high selection of quality roof. You make high selection of good bricks to put as a foundation. But then you're not thinking about what helps you in the actual window level of it. So you put shabby windows, people see your roof from afar, then they get to the building and like, oops, oh, it looked good from afar. So the planar cell polarity ensures that the middle of the cells also look good. And that impacts what happens at the top and at the bottom. So this is an important pathway where you have fungal and prickle in a complex. Now, let's focus on what happens here. Frazzled and DVL. Why I'm choosing to present fungal here is because the origin of fungal 1 research started with flies. So you have the strabismus in flies, which is important. So frazzled and DVL form a complex as well. However, the homeostasis of this complex regulates DVL activation, because once you activate DVL, then you activate the downstream effectors that lead to cytoskeletal rearrangements and migration. So if DVL is aberrantly activated, you begin to have dysregulated cytoskeletal rearrangements. So this is a core hallmark of cancer to look at, and that's what we did. Again, back to my favorite thing. So we looked at cancer cell lines, cervical cancer cell lines, and wanted to look at the dynamics of fungal 1 in those cells. We used a simple assay. Okay. We used a simple assay of RNA interference. I think you heard about it yesterday. So these are transient transfections, which means under 72 hours, you can read your result. And what we found is when you remove the oncoproteins, you have a chunk of fungal 1 rescued in different cervical cancer cell lines with HPV expression. And you also see that specifically with the cell expressing 16E7, the stability of E7 declines modestly. But now, how do we ensure that this is an onco-specific activity and not just a fluke from off-target? You bring in your controls. Just like Professor Yumi mentioned, the importance of having positive and negative controls in your clinical setting, even in research. C33 is an HPV negative cancer cell. And when you look at fungal 1, nothing is happening. When you look at the normal keratinocyte, which is the negative control as well, nothing is happening to fungal 1. So this is a full panel that shows me this is an onco-specific interaction. Check 1. Fungal 1 is important. The interaction is phosphodependent. Now, the dynamics differ from cervical cancer cells compared to non-HPV present. So we've established the baseline. Now what is next? So fungal 1 is a target. What does it do? Now we go to the functional assays. When you work with pathogens, you know that they do two things. They either reduce or increase a protein for them to have their way. So if they are not downregulating it, they are overexpressing it. And one assay to find out what is happening to your proteins in the presence or absence of a target is what you call the Chase assay, cyclohexamide assay. It's a very simple assay whereby you just add cyclohexamide, and then at different time points, you look at your protein level. And what we found here is that, interestingly, when you remove the oncoproteins, it's like you switch fungal 1 back to its original turnover time, because every protein goes through a natural turnover. So when we look closely at what was happening to the control, you see there are two bands here. The top band is phosphorylated fungal. The lower band is unphosphorylated fungal. That has been confirmed by our lab. And you see that while in the SRA67, there is a steady concurrent decline. Both bands are going down in similar patterns. But in the scramble, you have this sharp decline and it just stays there. So we know there is an aberrant or defective turnover. How do I know that? Because the loss of the oncoproteins does not deplete or reduce the natural turnover of fungal 1. What is the next question? Actually, we repeated this assay so many times because when you work with HPV, after some time, your mind is conditioned to thinking plus or minus. It must be rescued. It must be degrading it. But this wasn't the case. It was a non-canonical dysregulation. So how do you investigate defective turnover? Another mindset shift. We decided to go in the way of, well, that was a reconfirmation in overexpression. The first one was endogenous condition. The second one was overexpressing the proteins and see what was happening to the dynamics. And what we found here was, interestingly, not only is VANGL1 sustained, but E7 is also sustained. So sometimes virulence factors or oncoproteins from pathogens interact with these host substrates for their own stabilization. They want to be able to stay there for as long as they need to be there. So when you have E7 alone, the half-life is less than 2 hours. But when you have it with VANGL1, you still have a good chunk of expression even after 8 hours. So that shows you that this interaction is extremely oncogenic. But what is it doing? If it's not a classical story of degradation, what is happening with mislocalization? So when you move from one data to another, I was speaking with somebody yesterday and they were talking about PhD, or was it this morning? And I'm like, sometimes your proposal and what you submit as thesis, they are not even relatives. You know, they don't even belong in the same family. Yeah, so if you want to condition your mind to follow strictly objective one, I would check phytochemical. What if you check the phytochemical and is like, what happens? Does the research end there? No. Your data must be able to speak with you and help you in the direction to go for the next thing. So we decided to check subcellular fractionation. Simple assay again. Pick your cell and divide fractions of it to check that protein. So instead of checking the whole cell lysate, you're checking the membrane fraction, you're checking the cytoskeletal fraction, and looking at the presence of your protein. What does that tell you? It sort of shows you a crude way of monitoring the trafficking of the protein and possible mislocalization. If my protein is supposed to be at the membrane, then I realize in an oncogenic situation, I'm finding it in the nucleus. It's moving away from the site where it's supposed to be functional to a site where it is defective. So here we found that the cytoskeletal pool of VANGL1 that is supposed to maintain the network with actin at the cell-to-cell junction is depleted in cervical cancer cells. And that was interesting because when you now remove the oncoproteins, it is restored massively. So not only was this really intriguing, we wanted to visualize that. You've seen it biochemically. How about you put some colors with IF? I'm mentioning all these things because during the course of the week, you'll be introduced to all these techniques. But why do I need it? Is it to make the paper bigger? Or is it actually to answer some questions? Because I think these days was, in my work, I have to put whole genome sequence. Do you need it? So instead of using this research and maximize it, you want your paper, somebody to pick your paper and literally have no idea because you've packed techniques rather than answering research questions. Now, visually, we saw that when VANGL1 is existing alone in its chilled space, you see a nice definition in the cell-to-cell junction, maintaining those cell-to-cell contacts. When E7, my favorite guy, is alone, it's predominantly nuclear, and you have some traces in the cytoplasm. Now let's put both of them together. First, you see that VANGL1 becomes to appear diffused. Exactly. So you begin to lose the integrity at the cell-to-cell contact. Does it correlate with that or not? It correlates with what you saw biochemically. And now what is happening with E7 is even more intriguing. E7 was appearing, casually in the cytoplasm. But when it has VANGL, it's happier. So it needs VANGL1 to buffer it in the cytoplasm. Now let's focus on these two cells. Now E7 moves from the nucleus to the membrane in certain cells. So now it's helping its shuttling by binding to VANGL1. Now you've answered the key question. If you've always been wondering, how does E7 move around and interact with all those substrates? It's because there are anchor proteins that bind to it for it to do its work. The questions you can ask in your research. Now, does this have any functional impact? Bangle 1, okay. Bangle 1 is important in migration in cytoskeletal rearrangements. How does that relate with cancer? We know that unchecked proliferation, and when you begin to talk about metastasis, that is the core signaling pathway that you're looking at. So a simple test again called the invasion assay. You have your matrigel on the membrane, introduce your cells to it, and you put food at the bottom for them and see how hungry they are to invade and look for the food. Then you begin to see that when you knock down Bangle 1, you move from this amount of invasive capacity to this level, which is almost shutting it down. So you lose invasiveness, but this is 2D. When you're studying polarity, especially in the plane, in a monolayer culture, you might not be capturing the right thing. The use of right models to answer the right questions. So we brought in the 3D models. And in these 3D models, again, we removed the oncoproteins in some and removed VANGA in some. And you see that when you remove the oncoproteins, you begin to experience significant structural disintegration. You see that with oncoproteins removal and that with Bangle 1. And I usually like to have a fluorescence or a 3D reconstruction of my sample so that you have a clearer view of what's happening in the tumor environment. And now what happens when you put this in invasive platforms? In this case, we used a matrigel collagen. You know that the tumor environment changes with tumors, right? You begin to have increase in collagen density. What does that do? Any ideas? Okay. The tighter the collagen, the more efficient the formation of fibers that go into it. So you have higher invasive capacity when the collagen concentration around the tumor increases. So how do you recapitulate that? You vary your collagen density. And that's what we did. And you can see that with increase in collagen, this tumor becomes actually much more aggressive. And you have a complete dissolution of even the spheroidal core, right? But when you remove the oncoproteins, you lose invasiveness. Check. When you remove Bangle 1, two things are happening. First, you lose invasiveness. But secondly, you have this distinct change in phenotype in the soft matrix. That is telling me there is a mechanical cue happening there. Then as you progress with this is even much more obvious that in the softer matrix, this guy changed from the spherical structure to this very funny amoeboid shape with loss of, so what does this mean? Next research question. Now, I want to visualize Bangle 1 distribution when the spheroids are invading, because that is where I think my answer is. If you look at the white type, which is invading, then you see that Bangle 1 is enriched selectively at the invasive fronts. So Bangle 1 moves to lead the cells that are invading. So it's core in collective invasion. What happens when you lose the oncoproteins? Of course, you begin to see a partial restoration of the localization. And now it's even across the rim of the spheroid. Now, what happens when I remove Bangle? Not only do you expect the loss of the signal because there is a knockdown of the protein, but you can see that we have two different panels here for one spheroid. And that is because the microscopic view, the spheroid was too large for it. Initially, we thought it was just a technical fluke, but you repeat and repeat again, and the phenotype is the same. So what is this saying? Then you look closely at the DAPI. The number one thing that jumps out here is there is nuclear crowding. If you look at the nuclei, they are overpacked, almost stacking on each other. What does that tell you in cancer? Always feel free to go back to your literature. What is the implication? A simple Google search of nuclear crowding. When cells that are usually invasive lose their capacity, for instance, somebody who likes to be out and there, and now the person has a stroke, what happens? They begin to pick up new habits. In the case of tumor cells, they begin to increase division as a compensation to try to survive. But then for every physical entity, there is a threshold. So when the capacity of threshold to accumulate cells is reached, the cells begin to induce the apoptosis and begin to die. So sometimes your answer is not in the actual thing, is the feedback from the target. understanding where to ask the questions and answer them. And I'm just going to round up quickly. I don't know if I should present this, but this back to that particular pathway, remember DVL activation. If you want to be sure that this pathway is actually activated, you go to the central switch. So we checked DVL2 in the samples and actually found that DVL2 is down-regulated when you remove VANGL1. And that tells me that the interaction of E7 with VANGL1 actually activates DVL. And that is what is responsible for the cytoskeletal arrangements and the invasive phenotype that we see. Again, simple questions to answer that. And to validate that, just go back to your invasive assay. Is it true? You move from Y type, take out VANGL, take out DVL2, and you lose invasiveness. So in summary, I won't go into this, but just know that you can also look at the chemotherapeutic part of things. Because from my 3D models, I can also test existing FDA-approved drugs. And from morphological readout, it can give you very cool information on how your spheroids are responding to that. So we did this with cisplatin, with etoposide, and just in brief, when you see all this disintegration and the loss of spheroidal integrity, is because they are dying off. And you look at the reconstruction, you see increase of cellular debris in the environment. So not only is VANGL1 important in the biology of HPV, it is also important in the treatment of it because it can inform therapeutic options. Now we have DVL antagonists, we have Rho inhibitors. So begin to think at the end point of all these resources, what does this achieve? Who does it benefit? Do we have existing drugs that are targeting this pathway, or am I developing it? If you're working on treatment with plants, with whatever it is you're dealing with as compounds, is it existing already in pure form? Rather than macerating mango leaves, mango bark, banana leaves, think about the active compound. That was my mind shift. I worked with peptides from spinach. So instead of cutting spinach and, 72 hours, shake in ethanol, why not just pick the peptide, synthesize it, or produce it in recombinant way and apply it? That way you move away from the crude way to giving people something closer to being believable. And then you begin to think about formulation. You'll be surprised how much it is or how easy it is for you to formulate a topical gel. It's because we are not thinking about it. Think about hydrogels. If I put this extract in an hydrogel, you've taken it away from just crude, because how many people do you want to serve spoons of moringa to, really? Even your family members won't take it from you. Even you, as the researcher, how many of your plant extracts have you taken at home? Good answer. So let's begin to have a change in a mindset. If you can't sell something to the investor because you don't believe it, because if I'm an investor and you tell me, oh, pawpaw works so well in curing typhoid, I say, okay, how many have you eaten when you had typhoid fever? The answer is nothing. So let's begin to have a mindset shift that you can achieve a conclusion that has pharmacological or therapeutic implication on what you do. And on that note, I would like to acknowledge my collaborators on this work, Mike, and that's Lawrence, my boss. And thank you. So I would like to know if you have any take-home message or any questions. Thank you so much for your attention.
So we have few questions online as well, but we'll take questions and comments from the house first. Thank you so much, Dr. Ife. Questions from the house? Great. I feel like it's more of a comment than a question.
Oh, okay.
And so to applaud you again. Because I mean, you started from like how you were on -0 or -100. Yes, and some days I still feel like I'm on that level. Yes, and just seeing everything that like the lab you're in has walked through, and just like at every step of the research, there's more ways to ask questions. It's not just linearly. Yeah, and I think Professor Ross mentioned it yesterday, how to look horizontally, not just linearly. And I mean, it's just wonderful. I feel like I still need to go over what you did again.
Thank you so much. Thank you so much.
So you can go back and rewatch. In fact, I advise you to rewatch at least, not at most 48 hours after the session, so that you will not forget everything before you rewatch it. Comments or questions from this?
Science in its simplest form. You have done well.
Oh, thank you. Thank you so much.
I will link up with you.
Thank you, sir. It's FOC.
You have baptized me. I've been working with herbs, and I even discussed with Professor Ross. But you just gave me a way out now, and I will run with it. Two simple questions. One, first, this island with IDG, is it a kind of an NGO that people can belong, a platform that people can belong, or something? I want to be part of it if possible. Okay. Then secondly, I know that HPV have vaccine. So have you, in the course of your work, interacted the vaccine with the development of all these pathways that we have looked at?
Thank you very much. Okay. So first, thank you very much for all the great feedback, and to you as well. Sorry, I didn't catch your name. Yinka. I will be lying to you if I tell you that I still don't feel at zero in certain settings. You know, sometimes you go to a conference, you see people's presentations, like, nobody gave me a memo to this. Like, when did we start doing this? But one thing I've done is to always dissociate myself from the bogusness of it. Sometimes people think complex is beautiful. Sometimes what you're just saying is the simple story, this increases. But then you see all this complex visualization, and you're like, wow, that's like 1,000 dots. Do not focus on trying to understand it, but listen to the in-between spoken words of the presenters, and then synthesize it at your own pace, in your own way. Then to your question, I Learn with IDJ is a free platform. It's run by me for now. But what I do is to just have a chat. I mean, we have a few members here. Just have a chat about molecular biology techniques. So I run it for a couple of months per cohort. And I try to keep the class simple so that we can have maximum interaction. So I teach from the molecular dogma, talking about each molecules, up to techniques. And then I leave that cohort to have a ripple effect on people. So I always welcome if people want to join me in teaching. Absolutely, that'll be great. We have classes for now. I'm on the 4th cohort, and we have classes every 2 weeks, and it's virtual. So it would be great to have you join us. Thank you. And to HPV, sorry, he has 2 questions. So HPV, yes, HPV does have vaccines, but they are prophylactic, which means prevention. Once the infection is there, it's pretty much useless. So for our models, we work with transformed cells. So it's already happened. It's cervical. So the part of vaccine is gone at this point. So we are thinking about treatment. So that is why we've not interacted with vaccination. But there are labs that focus on trying to establish the natural infection process. So those can also actually use, like John Dober's lab in Cambridge, they do a lot of infection studies with beautiful models. So you might want to follow their work and check as well. Thank you so much. Did I answer all questions? Okay, thank you.
Just a follow-up question.
Yeah. Yes. Thank you. One major problem we have with HPV vaccination is the coverage. It's very low in Africa. I was interacting with people yesterday. My notion is that HPV screening is free. But because I've always been picking.
So the thing is, for the vaccine rollout, there was a national rollout last year, and it was for girls aged 9 to 14 because they believe that they've not had their first sexual encounter. However, even for this, I was discussing with some people that there's a lot of misconception.
Absolutely.
And I heard the girl tell me that they told her that if you take the vaccine, then you won't have more than one child when you grow up.
Exactly.
So there's a lot of misconception around that vaccination, and that's why a lot of people are hesitant. In fact, me as someone who is older, when I go to the health centers and they say, no, pay for it, we can't give you for free, we can only give children who are less than 14
for free, 9 to 14 for free. So, you know, there's a lot of barriers when it comes to the rollout and implementation, especially at public health level.
Thank you so much for that, because I know you're actively on the field. And also in Africa, it's still majorly for girls. In EU, for instance, it's boys and girls because it's not a one-way thing. So when it comes to vaccination, we still have major issues there. Yeah, and hopefully we can change that.
It's cheap. It's not free, and I don't even think it's cheap.
I was surprised to hear that yesterday.
Thank you very much for the awesome presentation.
Thank you.
I just had a little bit of a question. You mentioned clathrin-dependent endocytosis.
Yes.
AP2M1 and E7.
Yes.
And on slide 26, when you were talking about the collagen density and the protrusions of those, so it made me remember or think of Philopodia and Lamentipodia. And when you were talking about metastasis, that's when I just blotted out a word. So my question is macropinocytosis is indicated in the metastasis of these? And some of the structures on the pictures you showed, they appear to me as though probably I was asking, did you actually work with organoids or something like that? So that's like, yeah, those two questions.
Okay, so you were asking about macropinocytosis. Well, for HPV, it is one of the endocytic machineries, but it's not the major. So we have the caveolin, we have the HSPs, the heparin sulfate glycans. We have macro, which there is a whole lot of body debating on that. But the clathrin is newly identified through AP2M1. So that's just a brief answer there. Then you were asking, yeah? So my work is not, I don't do internalization. Those are people who deal with entry. I'm always on the end part of things. Now it has entered, what happens? Yeah. So I can refer you to people like Justina, an amazing Polish lady who does a lot of work with L1, L2 antigens and what happens in the entry with SNCs and all of that. Yeah. Yeah. Okay. Then your second question was about if I used organoids. Those are spheroids. But then there's another part of the work where we can show you cervical organoids as well. Okay.
So we have some questions online.
Oh, wow.
So the first one says, since HPV stays silent in the body, does it mean if this is checked in an individual where it is not yet symptomatic, will it be detected?
Oh, absolutely. Oh, I mean, that's the importance of genetic testing. Because when it gets to that basal layer, it deposits its nucleus. It deposits its genome, sorry, in the nucleus of the host cell. So you would find it. And that's why DNA testing, genetic testing is the gold standard for majority of these things. If you're expecting to pick the antigen at that level, perhaps you won't see it with VIA screening, but with genetic testing, you will definitely pick it up. So the answer is yes on that. Asymptomatic does not mean it's not there. It just means it's not eliciting clinical symptoms that can send you to the doctor. Yeah.
Have you tried check this VANG1 in 3D? I think you've answered that.
Tried checking this. She has answered that already.
Yeah, the spheroids are also 3D, by the way. So they are 3D models. Yeah.
What are the strongest mechanistic and experimental data supporting a causal or contributory role of these pathogens in glioblastoma initiation, progression, or treatment resistance?
So can we slow down on that? What are the mechanistic
and experimental data supporting a causal or contributory role of these pathogens in glioblastoma initiation, progression, or treatment resistance?
In glioblastoma?
Yes.
Oh, wow. That's glioblastoma. I mean, this is cervical carcinoma. I'm sorry, I'm not trying to be silly. But okay, here is it. There are several targets that you find in one cancer that are also dysregulated in other cancers. So there is always a link. For instance, VANGAL is also dysregulated in glioblastoma, but it's usually context dependent. In some cancers, you might have overexpression causing the problem. In some, it might be lower expression causing the problem. So you can always take inferences from one cancer and see what is happening with that target in your own form of cancer. So I'm sorry, I would be happy to answer, but I know nothing about glioblastoma other than what I've just told you. Thank you.
Okay, one more question. Right.
Yes.
Okay, I know that the use of collagen model in 3D has a way of transferring virus to the cell. Do you try using the clinostat system where this cell can spontaneously form from the model?
Can spontaneously form what?
From the model. I don't think the question. Okay. I know that the use of collagen model in 3D has a way of transferring virus to the cell.
Transferring virus.
I don't know.
Honestly, I would have loved if the person was on site because I would love to learn that.
Okay.
It's a gray area for me, but I promise to read more about it.
Okay.
So
how can we distinguish true oncogenic involvement from opportunistic infection from tumor
okay,
tissue?
those are two layers: 3D model and distinguish opportunistic infection.
Yeah.
Okay, so I'm just going to answer this from my microbiology background.
Yeah.
Opportunistic means they are naturally not disease causing, but they fall in situations whereby the environment allows them to be pathogenic in nature. H. pylori is a clear-cut guy. He knows what he's coming to do. HPV is a clear-cut guy. He knows what he's coming to do. He's not coming to play with you. But when you begin to look at the gut, sometimes the gut microbiome contains possible opportunistic pathogens. I think one of the studies I did in Unilever that was on E. coli. Most times E. coli tends to become pathogenic. So in such situations, does it directly lead to oncogenesis? That's a big question mark. Can it stimulate changes that can downstream lead to oncogenic signaling? Perhaps. And what's the answer to that? Research. So that would be my answer to that. So thank you very much.
Let's give Dr. Ife a round of applause.